on proteins and amino acids. Proteins are just like carbohydrates and lipids, very important macromolecule. It's an important macromolecule required by the body and found in abundance on the earth. So now let's come to the question of what are proteins. They are very large molecules. They are one among the macronutrients. They are polymers of amino acids. That means they are made up of chains of amino acids. They are found in every cell in the body. They are involved in almost all of the functions of life's processes, right from hormones, digestion, your muscle action, everything involves a large number of proteins. In fact, the breathing, the oxygen that you take in and take out is through a protein called hemoglobin. And the proteins, as I said, are sequences of amino acids. And these sequences are not random sequences, but they are determined by your DNA. The DNA dictates what the sequence of the amino acid in a protein should be. So a protein is made up of, it is considerably simpler than what we had seen for lipids. It is made up of 20 different L amino acids. That is the configuration, the stereochemical configuration is L. So all the amino acids are L amino acids. And if you recollect, I said all the sugars that we use are D sugars. In the body, we know water is the most abundant substance, and protein comes in second. So, what is a protein and what is the structure? Proteins are classified as either as peptides or as proteins. In the peptides, these are polymers which contain less than 50 amino acids. They could range anywhere between 2 to 50. And when the number of amino acids in the chain are more than 50, then we call it as a protein, and they can range anywhere from 100 to 10,000 amino acids which are linked together. Proteins, as I said, the amino acid sequence or the way in which they are put are dictated by our DNA. And you know the dogma of molecular biology, the central dogma of molecular biology is that DNA, you have it's transcribed into RNA, and then from RNA it is translated into the protein. So for every amino acid, there is a 3 code, that is a 3 nucleotide code, which tells you which amino acid it is for. Amino acids are organic molecules made up of carbon, hydrogen, oxygen, and nitrogen. So, looking at the anatomy of the amino acid, just like fatty acids, the amino acid is an alpha amino acid. Here, the amino group, it got its name from the amino group. The amino group is linked to the alpha carbon of the amino acid, and therefore they are known as alpha amino acids. If you look at the alpha carbon of an amino acid, it is linked to 4 different groups. One is the carboxyl group, the other is the amino group, the third is a hydrogen, which is common to all amino acids. And then you have the unique side group R, which varies and gives you the 20 different amino acids. So in an amino acid structure, you will have an alpha carboxyl group, an alpha amino group, and an R group, which tells you which amino acid it is. So we have 20 different amino acids, and we will see the structure of them and how they differ. The simplest of the amino acids is glycine, where R is H. So glycine is the smallest amino acid, which has an alpha carboxyl, an alpha amino group, and two hydrogen attached to the alpha carbon. Then you have examples of aspartic acid and phenylalanine. We will see all the amino acids later. So what are the general properties of amino acids? There are 20 amino acids. All of them are alpha amino, or they've got the primary amino group, except proline. Proline has a secondary amino group. So instead of NH2, it has a NH. The carboxyl and the amino group of the amino acid have pKa's ranging. The carboxyl group is weakly acidic, so it is around about in the acid pH 2.2, and the amino group is weakly basic, and so it has a pKa of 9.4. At physiological pH, all the amino acids are charged as shown to the left. They can act either as an acid or they can act as a base. And because they can exist either in the negatively charged form or in the positively charged form, they are referred to as Zwitter ions. This is something new that you're hearing in the past 2 days. Neither a fatty acid nor a sugar can exist as a Zwitter ion, but an amino acid can exist as a Zwitter ion. And this imparts very special properties in that all the amino acids have got very high melting points, about 300 degrees centigrade, just like salts. And they are soluble in water. The reason being they have the carboxyl group, which is polar, and the amino group, which is polar, which renders them soluble in water, but they are insoluble in organic solvents. So in the amino protein, what is important is to know what is a peptide bond. So now you know the building blocks of proteins are amino acids. Now let us see how the amino acids link to form the peptide bond. The alpha carboxyl group of one amino acid will react with the alpha amino group of a neighboring amino acid. A molecule of water will be lost by condensation and a peptide bond will be formed. So the peptide bond forms by condensation when the carboxyl group of one amino acid and the amino group of the other amino acid join to form a peptide bond. Now the same dipeptide can react because it has got direction. It once again has got a free carboxyl group and a free amino group. This free carboxyl group can react with a neighboring amino group of a neighboring amino acid. It will lose a molecule of water by condensation, and then you will have a tripeptide. Now, when they can condense and form a peptide bond similarly can be broken by hydrolysis, either by acid hydrolysis or base hydrolysis. So if you take the dipeptide, the acid or the base will break this peptide bond and it will give you the free carboxyl, that is the free carboxylic group, free amino acid, and the other amino acid. So peptide bonds are made by condensation and they are broken down by hydrolysis. So the peptide bond forms by the elimination of water, and you can have di, tri, oligo, and polypeptides, and you have polymers of very large size also. So a small protein is of about 100 amino acids. Now, if you think of a protein with 100 amino acids and we have 20 amino acids, the number of ways in which the amino acids can be arranged in the protein will be in the order of 20 to the power of 1000, or it would be 1.27 into 10 to the power of 130 possible unique polypeptide chains. This would happen if we were doing it chemically. But since the sequence is dictated by the DNA, so when protein biosynthesis occurs, it is only the amino acid, the code, the mRNA for the particular amino acid with the code is taken, and that is inserted into the protein. That is the protein biosynthesis. But in chemical synthesis, if I have just two amino acids, glycine and alanine, I can make two peptides, because one peptide is gly-ala and the other peptide would be ala-gly. And the two are entirely different from each other, although they contain the same two amino acids. So coming to the classification of amino acids, in addition to the alpha amino acids, we also find some amino acids where the carboxyl group, we have the beta amino acids and the gamma amino acids. But you will not find these amino acids in a polypeptide chain. You can find them free. For example, a beta amino acid is beta alanine, whereas gamma amino acid, you must have heard of the neurotransmitter GABA, that is GABA amino butyric acid. You never find amino acids other than alpha amino acids in a protein or a peptide. So there are 20 genetically encoded alpha amino acids. Of them, 19 are primary amines. That means they contain the amino group, and one of them is a secondary amine, and that is proline. So since in the amino acid you find that the alpha carbon is attached to 4 different groups, it can exist as isomers, as stereoisomers. They can exist in the conformation of L or D, because this carbon is, this is alanine, this carbon of this is glycine. This carbon of alanine, if you look, has got would have 4 different groups attached to it, and so therefore you can have both L-alanine and D-alanine. Glycine is the only amino acid which does not have any chiral carbon and therefore exists only as glycine. You do not have D-glycine or L-glycine. So on this left-hand side, what you see is the structure of glycine, where you see carbon is attached to only 2 different groups, and this is common hydrogen. And so therefore, this carbon cannot become a chiral carbon. And from the carbohydrate class, you should remember that a chiral carbon is a carbon which has 4 different groups attached to it. And glycine is the only amino acid which does not have a chiral center. Now coming to the classification of the amino acids, other than them being known as alpha amino acids, they can be classified based on their side chain. That is the R group of the amino acid, and it is the side chain of the amino acids that are responsible for the unique properties of the proteins. There are 3 major side classes, side chains. One is the hydrophobic, that means amino acids which are having the hydrophobic side chain, like valine, isoleucine, leucine. You have the charged side chains, like that of aspartic acid, glutamic acid. You have polar, and you also have non-polar, and plus we can classify them as sulfur-containing amino acids, or we can classify them as carboxyl group containing amino acids, or we can classify them as basic amino acids. But The 20 different amino acids are commonly abbreviated. Earlier, they were abbreviated with a 3-letter code, but now nobody uses the 3-letter code. It is the single letter code which is used to represent an amino acid. Like A is for alanine, G is for glycine, for tryptophan it is W, for tyrosine it is Y, for methionine it is M, for aspartic acid it is D, for asparagine it is N. So all the 20 have different letter codes. So here is the example of the amino acids which are classified based on the non-polar nature of the side chain. So you have glycine, which hydrogen is the side chain. You have alanine, you have valine, which has this 3-carbon. You have isoleucine, leucine, isoleucine. These two are isomers. You have methionine, which contains a sulfur group in its side chain. You have proline. This you must remember is an amino acid. You have phenylalanine, which is aromatic. It has an aromatic ring. And you also have tryptophan, which has the aromatic side chain. So these would be also classified as aromatic amino acids. So in the polar, you have serine, which has a hydroxyl group. You have threonine, which has a CH2OH group. You have tyrosine, which is very similar to phenylalanine, but except that the benzene ring is hydroxylated. You have cysteine, which is a sulfur-containing amino acid. So you must remember the sulfur-containing amino acids. There are only 2 of them, methionine and cysteine. Then you have asparagine, which is the amide form of aspartic acid, and you have glutamine, which is the amide form of glutamic acid. We can classify them as acidic and basic because you see aspartic acid, in addition to the carboxyl, alpha carboxyl group, it also has another carboxyl group. So it has a side chain of CH2COOH, and this is CH2CH2COOH. So aspartic acid and glutamic acid are the acidic amino acids. The basic amino acids are lysine, histidine, and arginine. And the aromatic amino acids are phenylalanine, tyrosine, and tryptophan. The sulfur-containing amino acids are methionine and cysteine. And then you have another group which is in vogue these days, especially the bodybuilders, to build your muscle, called the branched chain amino acids. And this is the branched chain amino acids are isoleucine, leucine, and valine. So foods which are rich in branched chain amino acids are used for the muscle building, or when you go to a gym, this is what the food is. You're recommended to take proteins containing high amount of BCAA, that is branched chain amino acids. Now you looked at the amino acids and you see that it has an alpha carboxyl group and an amino group. Now because of having both an acid and a basic group on it, these amino acids can exist both as a negatively charged molecule or as a positively charged molecule, and they are known as Zwitter ions, or amino acids are amphoteric. That means they can exist either as acids or as bases. And amino acids are characterized by their isoelectric point. That is the point at which the positive charge is equal to the negative charge. So you have the pH at which the amino acid exists in both the negative and positive is known as the isoelectric point. So when the pH is brought to the acidic pH, when pH is lowered, you see it exists as a positively charged molecule. When the pH is increased to the basic pH, you see that it exists as a negatively charged molecule. So at one particular pH, it will have both the positive and negative charge, and that pH is known as the isoelectric point of the amino acid. So how do you calculate the isoelectric point of an amino acid? The isoelectric point of the amino acid is the pKa of the carboxyl with the pKa of the amino group and then divided by 2. So if you look at alanine, pKa1 is 2.3, pKa2 is 9.7. So put together, this is 12. So the isoelectric point is 6. So at pH 6, you will see that alanine can exist in this state. Similarly, you can calculate for aspartic acid. It's 1.9 and 9.6. So is the pKa2, but if you calculate the pKa for the acidic group of it, it is 3.6 plus 1.9 divided by 2, which is 2.7. And for lysine, you take the pKa of the low pH and you take the pKa 10.5 and then you divide it and you find that you get the isoelectric point, that is when it exists in this form, is 9.7. The pKa's of different amino acids are given over here. So what I want you to look at is to see that the pKa's of the basic amino acids are very high, whereas the pKa's of the acidic amino acids are very low. That is their side chain. So you see arginine 12.8, histidine 6.0, and lysine 10.79, showing that these are highly basic. On the other hand, you see the pKa of aspartic acid and glutamic acid. It is in the acidic range. In glutamic acid, it is 4.07, and aspartic acid, it is 3.9. The amino acid properties are varied, so therefore they can be grouped into so many different groups other than the major groups that I told you about. So you can also group them as the small amino acids. That includes glycine, alanine, serine, and cysteine. You can group them as sulfur-containing amino acids. Here you have the branch chain amino acids. Here you have the aromatic amino acids. Here you have the basic amino acids. Here you have the acidic amino acids. This is the single letter code for the using the single letter code, and I'm sure most of you will know or somewhere you can find the single letter code for the amino acids. K is lysine, H is histidine, R is arginine, this is tryptophan, this is tyrosine, this is phenylalanine, this is glutamine, this is glutamic acid, this is aspartic acid, and N is asparagine. Otherwise, most of the amino acid single letter code is the first letter of the amino acid. However, if the first letter repeats, for example, threonine, tyrosine, they both have T. So threonine is taken, T is given to threonine, and for tyrosine we give Y. So having known about the properties of amino acids and also how they are classified, you just also need to know what are the tests, because we can identify amino acids by certain color reactions. The universal test for amino acids is the ninhydrin test. That is, if you add ninhydrin reagent to amino acids and heat it all amino acids will give you a purple color, except proline and hydroxyproline, which will give you a yellow color. You will also see that if by chance you spill ninhydrin on your hands, you will see that it turns purple, showing that the skin on your hands is a protein, and so you get a purple color. Then you have a test called the xanthoprotein acid test, which can actually identify the aromatic amino acids. So here, when concentrated sulfuric acid, concentrated nitric acid is added, you find a yellow color, and that is because there is nitration of the ring. Lead sulfite acid is used to detect the sulfur-containing amino acids, that is the cysteine and methionine. Millen's test is used for amino acids containing hydroxyphenyl group, that is tyrosine. The Sakaguchi test is used to detect arginine. The Hopkins Cold test is used to detect tryptophan, and the Biuret test is used to detect all proteins. Now, coming to the structure of proteins, you know they were amino acids, and the amino acids can be built into polypeptide chains. Now, protein structure has got a hierarchy, and it is divided into 4 levels of structure. One is, there's the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. Any alteration in the structure or sequencing changes alters both the shape and the function of the protein. So the structure, this hierarchy of protein structure is very important for function of the protein or the functionality of the protein. So in brief, just let us look at the hierarchy of the structure. So there are 4 levels of structure, as you said. There's the primary, the secondary, the tertiary, and the quaternary. And we will look at each one of these. So the primary structure is literally the order in which the amino acids are there, or it is a sequence of amino acids, almost written like a very long word which makes no sense. But if you know the amino acid sequence, the amino acid, it does make. So you will not find this word in the Webster's dictionary, but this is a protein which has some function. So this sequence is determined by the inherited genetic information that it is given or it is obtained by the DNA sequence. Then we have, in the primary structure, it is basically the peptide backbone, what we call, or the peptide backbone, the peptide bond, NHCO. So you see this peptide has serine, tyrosine, alanine, glycine, and Lu. And the length of these peptides depends on the DNA sequence where the termination will occur. Now coming to the secondary structure, we have 3 types of secondary structure. One is called the alpha helix, the other is called the beta pleat, and the third is called a loop, which allows the protein to bend and fold. So what is this alpha helix? What is beta pleat? And what is the loop? So you look at the alpha helix, because this structure, it took a very long time to determine this structure. It was determined by Pauling and Corey, and it was Max Perutz who found alpha helix in many proteins. So when you see an alpha helix, it is nothing but turns. But these turns have a definite interval. So every turn of the alpha helix is equivalent to 3.6 residues. So how this happens is the carbonyl in each carbonyl is hydrogen bonded to the NH, which is exactly 4 residues away. So if we go back and we see this primary structure, in the alpha helix, the CO of serine will be in hydrogen bond with the amino acid, 4 amino acids away, that is glycine, with this hydrogen. So there will be a hydrogen bond between this oxygen and this hydrogen. Next, there will be a hydrogen bond between this oxygen and with this next amino acid, that is leucine. So every carbonyl oxygen will be hydrogen bonded to the NH of the fourth, that is fourth residue from there. So you have 4 residues away. So therefore you have exactly 3.6. It is equal to 3.6, but we say because you cannot break a residue into half, so you have a turn every 3.6 residues. It is more like a telephone wire. If you have looked at the old BSNL telephone wires, I don't know how many of you have seen such wires, because I'm sure most of you use mobiles and there will be no wire on it. But if you can go back home and look at your telephone wire, you will see this as an alpha helix. And if you allow it to fall down, you will see that the turn changes, distance changes. But in a protein, the distance does not change. It remains uniform throughout. So the alpha helix, it is the most abundant secondary structure in a protein. Every turn, there is a turn at every 3.6 residues, which is about 5.4 angstrom. So the carbonyl oxygen forms a hydrogen bond with the NH of residue I plus 4. This is called intrastrand hydrogen bonding. All the NH are hydrogen bonded except the two ends, which will remain unbonded. And so therefore the alpha helix is known as amphipathic, because it has got a positive charge and a negative charge. So you look at hemoglobin, the protein which carries oxygen, you see that it is made up of a large number of alpha helices. Now coming to the beta pleat is the second major structural element. It is known as a beta strand. It generally is not more than 5 residues long. They can be parallel or anti-parallel to give you this pleated appearance, and so therefore they are known as beta pleats. So unlike alpha helices, these are formed in different parts of the sequence, and the hydrogen bond is between two protein strands. It is not within the same strand. In alpha helix, you see that every first amino acid is in hydrogen bond to the residue 3.6 residues away. Here, they are parallel, it's between two strands, so the hydrogen bond is between the carbonyl of one strand with the NH of the other strand, which you will see. So here is a beta pleat, so they're very short, between 5 to 10 residues. This is parallel because the strands are running in the same direction, and when they are it is between the strands, so you have the hydrogen bond from here to here, from here to here, from here to here, from here to here. That is the carbonyl oxygen and the nitrogen, the hydrogen attached to NH. The red is oxygen, and the blue, if it is visible to you, is the hydrogen on the nitrogen. So this is a parallel beta sheet. Now, if the arrows are in the opposite direction, it is known as an anti-parallel beta strand. So here you have the anti-parallel beta strand. The hydrogen bonds are very similar, but they are in opposite directions. So you have the alpha helix and you have the beta pleat. So if you look at the protein nucleoplasmin, it is mainly a beta pleated protein structure. Now coming to the tertiary structure, once you have the alpha helix and the beta pleat, these proteins will fold over one another, and uh, they will form a globule-like structure to form a globular protein. And this is possible by three different interactions due to ionic interactions of the side chains. So if you have a carboxyl group of aspartic acid, it can form a hydrogen ionic interaction with the amino group of lysine. So that is called an ionic interaction between opposite charges, or it can form hydrogen bonds, which are very weak bonds, or it can form van der Waals forces, that is hydrophobic residues, hydrophobic interaction. So tertiary structure is the folding of the protein upon itself. So this is how visualize the protein structure. So it is the side chains of the amino acids which are pointing out they will interact with each other. So here is a van der Waals or a hydrophobic interaction with the side chain of isoleucine. You can have a hydrogen bond between the hydrogen of a serine residue with that of a carboxylate of an aspartic acid residue. You can have an ionic bond between the carboxyl group of an aspartic residue with the amino hydrogen of a lysine residue. So this is an ionic bond, a hydrogen bond. You have van der Waals forces or the hydrophobic bond, and you also have what is known as a disulfide bond, where the sulfhydryl group SH group of cysteine can easily be oxidized to give you the disulfide bridge. And I'm going to give you an example of everyday example that many of you girls, if you are in the audience, use when you go to the hairdresser. So proteins based on their tertiary structure are divided into two. One is the fibrous structure, which is elongated. Examples of the fibrous structure are your hair, silk, keratin, collagen, even the bird's beak, claws. These are all fibrous protein. Even your nails are protein. And then you have the globular proteins, which are globular and uh, easy to think, and the globular nature is required for them to express their functionality. So most of the time, the fibrous proteins actually are the structural proteins. That means they give structure, like the hair, the collagen, the keratin, the beak, your nails, and the skin. These are structural proteins. Now, globular proteins are proteins which do function, the functional proteins such as the enzymes, the hormones, the carrier proteins like hemoglobin, transferrin. So all these are globular proteins. To show you the structure of a single hair, this is what one strand of your hair looks like. What this is basically, it is the basic unit is a alpha helix. Now, two alpha helices will be coiled one on top of the other. So you have two alpha helices coiled, and then you have two of the two coils which coil upon one another. Then you have four of these protofilaments which will be hydrogen bonded and coiled upon each other, and you have a large number of this, and so you have hair. So that is why hair is so strong, because it is so strongly hydrogen bonded by the different alpha helices. The keratins in mammals, you have the alpha, all mammals have alpha keratins, and you find the beta keratins in reptiles and birds. So now look at what happens to your hair. See, when you wash your hair, what you find is it straightens. But however, when it is blow dried or you dry it back, it comes back to its original straight state or permed state. Why does that happen? So what happens in dry hair is it is in the helix is an alpha helix, and the keratin is in called the alpha keratin. So you have the hydrogen bond. So this is your dry hair or the unstretched hair. Now, when water is added and your hair is wet, you will find that the hydrogen bonds are broken. And when hydrogen bonds are broken, the wet hair is stretched, it will become elongated or long. However, when it is dry, blow dried or thing, the beta keratin, it'll it'll go back into the unstretched form. So it goes from the stretched, unstretched to the wet hair which is stretched, and then again when it dries, it'll go back to the alpha keratin state. So wet hair stretches as the temporary hydrogen bonds are broken, and when dried or the blow dried, they will return back to the alpha keratin. How many of you girls who are in the audience go to a beauty parlor to get your hair as permanent curls or to straighten your hair? All this also involves protein chemistry. I don't know how many of you have ever thought that it involves some science, that is, the perming of hair, making curls, breaking curls, straightening the hair. So just let us see for interest what happens. So in a hair, in addition to the hydrogen bonds which are there in the alpha helix, the hair is held together by these covalent bonds, the disulfide bonds. So that is cysteine is there. So cysteine of one helix will react with the cysteine of another helix, and you have the disulfide bridges. So this causes the curling, or what we call as the permanent perm in your hair. So when you go to the beauty parlor, what is done is first your hair is wet so that the hydrogen bonds are broken. Then they spray a reducing substance, mostly ammonium thioglycolate. So what ammonium thioglycolate does is it breaks the disulfide bridges. So no longer these disulfide bridges are there. So it will be straightened, or it will be put into curlers or rollers, and then it will be sprayed with an oxidizing agent. So when it is sprayed with an oxidizing agent, the disulfide bonds will reform, but maybe in different places, and what you get is a different perm or a different curl from what you had originally. But remember that this perm or curl will be there as long as the new hair doesn't grow out. As soon as the new hair grows out, you will have the original structure. You cannot change the structure. And so that is the reason why you have to keep going back to the beauty parlor or to the hairdresser to get your hair straightened or curled. Now coming to the quaternary structure, this is how the globular protein or thing is arranged in space. So these are what we call as the subunits. So proteins can be single subunit proteins. They could be multiple subunit proteins. So for example, you look at hemoglobin. Hemoglobin is made up of two alpha chains and two beta chains, and it forms a tetramer called alpha 2 beta 2. and each chain binds one iron molecule. So it carries a large amount of iron. So this is the example of hemoglobin. So you see hemoglobin is made up of 4 different subunits shown to you in different color. So quaternary structure tells you the way the protein is assembled, the number of subunits you have. Myoglobin, which is found in the animals, in fish, whale, and thing, is a single subunit protein which carries oxygen, whereas hemoglobin is a tetramer which carries the iron. So what happens to a protein when it is denatured? So the alpha helix is broken, the beta pleat is broken, and the globular nature of the protein is destroyed. So this is a normal protein which has alpha helix, beta pleat, and it is folded. And when it is denatured by heating or by adding an acid or by salt, you see it becomes a single chain. It comes back to its primary structure and known as a denatured protein. The best examples would be for you to look at is the egg white. When you break open an egg, you see a gelatinous, translucent egg white because the albumin, the protein in egg white, is a globular protein. However, on cooking or heating, you see it becomes translucent, it becomes opaque rather, and that is because the protein is denatured. Even paneer, the protein in milk, is held in solution, but when you add lime juice, you're bringing down the pH, you're breaking down the globular structure into this. The advantage of the denaturation of proteins is the digestion of proteins uh, is improved in a denatured protein rather than in a globular protein. And the digestion of fibrous proteins is much harder and it doesn't take place. And that's why if you bite your nails or if you have the habit of chewing your hair, you will find that these proteins come out undigested because the fibrous proteins are not digested by the human body. So now that completes about the structure of proteins, but what is more important is to know about the nutritional quality of the protein. So proteins are of very high quality, they are digestible, uh, they are known to be high quality if it is digestible, if it contains all the essential amino acids, and if it provides sufficient protein to synthesize non-essential amino acids. So to know whether the protein that you are consuming is of high quality or no, it helps to be aware of certain statements and definitions. That is the essential amino acids, which are the essential amino acids, the amino acid score, the limiting protein, the protein digestibility corrected amino acid score, PDCAS, which is the gold standard for the nutritional quality of protein, the biological value of the absorption and retention of protein for use. So what defines the nutritional quality of a protein? It is the essential amino acid content and the digestibility of the protein. And we have just seen the essential amino acid content can be got if we know the primary structure, that is the odd number of ABCD of proteins. And the tertiary structure is the folding of the protein. We know that globular proteins are difficult to digest, whereas a denatured protein is more easy to digest. So for this reason, we classify the amino acids in an entirely different way, not based on their structure, but whether the human body can synthesize them or not. So you have the non-essential amino acids, which can be synthesized by the human body, and you have the essential amino acids, which are not synthesized by the human body. And it is very essential for you to know all the essential amino acids. That is leucine, methionine, isoleucine, valine, threonine, histidine, lysine, tryptophan, phenylalanine. The non-essential amino acids, all of these are synthesized in the body by different reactions. So some of the non-essential amino acids, for example, phenylalanine becomes essential in certain conditions, like for example, in uh, in phenylketonuria, tyrosine becomes essential because phenylketonuric patients cannot convert phenylalanine into tyrosine like ordinary individuals because of a genetic deficiency. They cannot, the enzyme required to hydroxylate phenylalanine is absent, and there is an inborn error of metabolism. And we will see more in detail some of the inborn errors of metabolism. So please remember all the essential fatty acids names. You must remember the essential fatty acids names because you get a large number of questions asking you to name the essential fatty acids. Then the quality of a protein is known by whether it is a complete protein or it is an incomplete protein. A complete protein is a protein which contains all the essential amino acids in the right proportion relative to the need as designated by WHO and FAO. WHO and FAO have designated the essential amino acid need according to the age. So they have given the requirement for 2 to 4-year-olds, 10 to 12-year-olds, and for adults. Also, a different requirement is there for pregnant and lactating mothers. Examples of complete proteins are meat, dairy products, that is milk protein, cheese, paneer, and soybean products. In the case of an incomplete protein, this is a protein which lacks one or more of the essential amino acids. So it could be lacking in lysine, or it could be lacking in methionine, or it could be lacking in arginine, or it could lack in 2 amino acids. So most of the plant-based proteins are incomplete proteins. And so therefore, they cannot serve as the sole source of protein in the diet. And an incomplete protein has low biological value, but the complete protein has got high biological value. Now you will ask me, the question is, what do the vegetarians do? And in India, the majority of the population are vegetarians. And if you look at a complete protein, soybean is the only one which is almost equivalent to the dairy and the meat. So the non-vegetarians will have no problem if they eat meat, but vegetarians have to get all their protein sources from paneer, from milk, and the other dairy products. But if you look at our dietary habit in India, you will find that our diet, the way we eat, we will have a complete protein by what we call a procedure as complementarity. So this happens in the Indian diet. So what you see is we eat either dal chawal or we eat dal roti, and we will see. If you look at wheat and rice, these are deficient in the amino acids methionine and tryptophan. But whereas the legumes as such, they are rich in the amino acids and deficient in the in 2 amino acids. But when we take a combination of the two, so you look at the legumes, it gives us plenty of isoleucine and lysine. They are very rich in lysine, whereas wheat, maize, they are all lacking in lysine, and they are very high in the sulfur-containing amino acids such as methionine and the aromatic amino acid tryptophan. So now if you eat dal and roti, what you have is you have a complete protein, and this we call as complementarity, complementing the proteins or their complementary proteins. So you can make up for the absence of an essential amino acid by combining the proteins and having them together. So that is why in the Indian diet, although the vegetable proteins are incomplete, the way we combine and eat it, you get a complete protein. Then you have fast versus slow digestion of proteins. So proteins are digested at different rates, and similar to the glycemic index concept of carbohydrates, we have got slow digesting and fast digesting proteins. So you have whey protein versus casein. These are two proteins which are present in milk. You will see that whey protein is rapidly digested, whereas casein takes a very long time to digest. So if you look at the composition of the human milk or the breast milk, you will find that the breast milk contains more of the whey protein and less of the casein protein. And that's why the young infants are able to digest the protein in mother's milk. So this is the reason also it has been said that you should not give cow's milk or anything to infants who are below 6 months, because it is very difficult for them to digest the protein of cow's milk, in which the casein is much higher. Soy protein is digested faster than whole milk protein, but the digestibility of the protein depends on the denaturation. If you denature the protein, it is digested very fast. That is because the enzymes can reach every nook and corner of the protein. Now coming to the protein quality, PDCAS is the gold standard, which is the protein digestibility corrected amino acid score. And earlier, the protein efficiency ratio was taken as the gold standard, but the protein efficiency ratio did not take into account the digestibility. And so therefore, this new standard of PDCAS was brought in by WHO and FAO to actually define the quality of the protein. So a protein that meets all the requirements for the essential amino acids of the body is given a score of 1. So let us look at the so if we look at the PDCAS values, you will find that milk has a PDCAS of greater than 1, milk protein, casein and whey protein. Soya protein is 0.95. You have all the meat proteins which have a PDCAS of 1.0. Among the plant proteins, it is soya protein which has the highest PDCAS of 0.95, which is almost equivalent to that of thing. So how do you determine PDCAS? So it has to be done by determining the amino acid composition of the protein. This is a very long drawn out process which takes almost 48 hours to make the sample prep, where the protein is first again hydrolyzed back into its amino acids, into the amino acids which made it. Then you have to derivatize those amino acids, separate them by chromatography, and then calculate the amino acid composition. And the hydrolysis for amino acid composition involves digesting it for 24 hours at 110 degrees Celsius, and even then not all the bonds in the between the peptides are broken down to give you the free amino acids. Several problems are there. Bonds between valine and isoleucine rarely break down. Serine and threonine will get destroyed. Methionine is only partially oxidized, and tryptophan is completely destroyed, and so also cysteine. So this is a long drawn process by which the analysis has to be done, and it is a very specialized analytical test, that is to do the amino acid composition. So this is just to show you, after hydrolysis and you separate it by HPLC, all the amino acids can separate, and then based on the standard, and if this is your sample, you can calculate the amino acid in your protein. So what happens to the protein when you take it by mouth? So this is generally digestion of the protein takes place. So first, essentially in the mouth, nothing happens to the protein because there is not a single enzyme which can break down the protein. So your food is just made into a bolus and you swallow it. When you reach when the food reaches the stomach, you see the stomach has gastric juice, which is acidic, and also it produces hydrochloric acid. So if you recollect in the past few slides, it said the protein gets denatured depending on the acid. So the hydrochloric acid in the stomach will denature the protein. So the globular protein will open and it will get denatured. We have the enzymes pepsin, which is an enzyme which acts only upon proteins. So this protein is broken down into a polypeptide chain. And then the food passes to the small intestine, where in the small intestine, the pancreatic juice comes in. The pancreatic juice contains two proteases or enzymes that can break down the protein, that is trypsin and chymotrypsin. So trypsin and chymotrypsin break down the proteins further into peptides, into tripeptides, dipeptides, and to single amino acids. And this, when it moves to the small intestine, the single amino acids are absorbed by the intestinal walls. The dipeptides are acted upon by dipeptidase, the tripeptides are acted upon by tripeptidase, and you get single amino acids which are absorbed into the body. So any protein that you take is finally broken down into the amino acids before it is absorbed into your body. So the absorption of amino acids takes place in the small intestine. Once it is absorbed, they are transported to the liver via the portal vein, and in the liver, the amino acids are used to synthesize new proteins. Some of the amino acids are converted into to give you energy. Some are converted, go into the metabolic pathway of carbohydrates, and they are called the glucogenic amino acids, and some are released into the bloodstream and transported throughout the thing. Occasionally proteins are absorbed intact, but when they are there, you have what is known as allergy. So what is the metabolic fate of the amino acid pool that is absorbed by the large intestine? So what you have is, you have the amino acids can go into gluconeogenesis. That means they can be taken up to make glucose when glucose is limited. The amino acids can be used for energy when the diet is deficient in kilocalories. So we know that when 1 gram of protein is broken down, you get 4 kilocalories of energy. Amino acids can also be converted into fatty acids, and the fatty acids converted into triglycerides, and these triglycerides are stored in the adipose tissue. In the body, protein turnover takes place. That means protein is continuously made and broken. So that is known as protein turnover. The amount of protein made should be equal to the amount of protein degraded. So a number of proteins in the body which have got important functions and are the enzymes are proteins. So they function to hasten the reaction. All enzymes are proteins except the ribozymes. The proteins can behave as defense proteins, that is the globulins, the immunoglobulins, the antibodies which are there, they are proteins. Proteins have a function of transport. So hemoglobin, the iron-containing protein, transports oxygen to the lung. You have other proteins like ferritin, which transports iron. You have the copper transporting proteins. So proteins are involved in transport. Proteins are involved in storage. For example, in egg, the protein is stored as ovalbumin. In milk, it is stored as casein. So they can serve as storage proteins. Then you have a large number of enzymes which are used, which are proteins, and they are used in the food industry. And we will be looking at them in detail when we have the webinar on food enzymes. So important to the food industry is rennet. You have fresh versus canned pineapple. The enzyme, pineapple enzyme bromelain, has got a lot of pharmaceutical applications. If meat doesn't cook in your home, you can add a raw papaya or you can add pineapple. It will tenderize the meat or break down the protein. Blanching of vegetables is done to inhibit catalase and peroxidase. So we will look at all these enzymes when the webinar on food enzymes is there. So proteins are also hormones. So insulin, which is very important for controlling the blood sugar and maintaining the homeostasis of sugar in the blood, is a protein. You have a number of receptor proteins. They are all these receptors are proteins. You have the contractile and motor proteins such as actin and myosin. So this muscle tissue, the expansion and contraction of your muscles or the motor thing are all proteins. And you have the structural proteins, which are the skin proteins, hair, horns, nails, fiber, etc. So just let's look at one or two proteins that are very common. This is collagen. Collagen is there. All your skin is collagen. Your ears made of all the connective tissue, that is such as bone, teeth, cartilage, tendon, ligaments. They are all made of collagen. Collagen is different from other proteins, that it doesn't contain the alpha helix or the beta pleat, but it has its own structure, what is known as tropocollagen, which is three intertwined chains of about 1000 amino acids, which are in an alpha helix form, which is different from that of what we have as the hierarchy. So we have 10 types of collagen, and each collagen has a different type of structure. But what you need to know is that collagen is a protein and it has a triple helix, which is known as tropocollagen. Those of you who eat gelatin, have you wondered what is the base of gelatin or jello that you use? It is nothing but denatured collagen. So any food that contains gelatin has to be put with a red dot, because gelatin comes from animal source. So that's why you have the vegetarian gelatin, which is the made from plants, that is the alginate, etc. Silk is a protein, and even the spider web is a protein. So all these insects produce silk. They make their cocoons, they make their webs, they nest in it also. So silk is a fibrous protein which is made of fibrin and a gummy protein sericin which holds these together. So the silk that you have seen is long stretches, okay, of 6 residues apart, or alternating of glycine and alanine. So the actual structure here is glycine, serine, glycine, glycine, and this keeps repeating. And then you see that they are hydrogen bonded between each other, and so therefore it is very difficult to cut the silk vertically like this. But however, because of the hydrogen bonds here, these can be actually elongated. So silk is a protein, but it is a protein which is different from that of the other proteins that you see in the human body. So best sources of proteins given to you are, you can see, meat has the highest amount of protein. So these are very high. Milk, the amount of protein is given. So the cereals and grains and vegetables have very low protein content. The protein content of wheat is about 12 to 14 grams per 100 gram. For rice, is about 8 to 12. So uh, the dals, that is the legumes, have much higher protein content. They range anywhere from between 22 to about 30%. So eating again grains and beans will give you your required protein content. So how much of protein do you require per day? Okay, so now comes to the macronutrient makeup for a healthy diet, telling you how much of fat, how much of carbohydrate, and how much of protein is required. So, as per NIN guidelines, that is the recommended daily allowance by the National Institute of Nutrition, which comes under the Indian Council of Medical Research, majority of your daily calories should come from carbohydrate rich foods. So for your diet, about 45 to 65% of the calories must come from carbohydrate, about 20 to 35% must come from the fat, and about 10 to 35% from protein. So majority should come from carbohydrate, especially if you're a individual doing hard work. Fat intake should be no more than 1/3 of the daily calories, and protein should provide the rest of the daily calories. So what is the required calorie for a normal individual? So for the normal individual, the requirement of the daily calories is about 2000 to 2200 calories. So what is the protein requirement? So what is it that you need to take? So normally for an adult, and I'm sure most of you are above 18 years of age, you need approximately 0.8 grams of protein per kilogram of body weight. So if you're a 80 kilogram individual, your diet should be about 64 grams of protein. If you're a 60 kilogram individual, it should be about 48 grams of protein. But what is happening is that because of this bodybuilding and getting the 6 packs or the 8 packs, most of the intake of protein is being misused in the form of protein intake for muscle buildup. So if you look at a sedentary individual, it is about 0.8 grams. For athletes, who do long distance running and swimming, you need about 0.97 to 1.37. So if any of you have heard or recollect, Michael Phelps, who was the swimming champion working, he would eat for breakfast an omelette made with about 10 to 12 egg whites. So that's the amount of good quality protein he would take. So for pregnant and lactating women, it is recommended that they take 30 grams per day. You will find these requirements in the recommended daily allowance. There is a book by NIN. It is called the nutritive value information and recommended daily allowance. And so that gives you what is the recommended daily allowance, and it also gives you the nutritive value of the Indian foods. So if you get a chance to get those books, you can find out how much of protein that you are consuming. So how do you do protein estimation? And I'm sure most of you know how to do protein estimation. There are two methods most commonly used in all analytical laboratories is the Kjeldahl method. So what does the Kjeldahl method involve? The Kjeldahl method involves, it is a three-step method where you have a digestion step, where you're breaking down the whole food into carbon, nitrogen, and oxygen. That is, all the nitrogen, organic nitrogen, is converted into the ammonium form. So you do a digestion with concentrated sulfuric acid. So when you do with the concentrated sulfuric acid, all of the protein is broken down and converted into ammonium sulfate. In the second step, you do a distillation where the ammonium sulfate, by a double decomposition reaction with sodium hydroxide, liberates ammonia, and this ammonia you titrate. So the ammonia is vaporizes. So because it is vaporizes, you have to trap the ammonia. So what you do is you trap the ammonia in boric acid, or you capture the ammonia in boric acid, and you get ammonium borate. And this ammonium borate is back titrated with hydrochloric acid. So for those of you who did not know the basis of the Kjeldahl reaction, so it is degraded. So all of the nitrogen is first converted into ammonium sulfate. Then the ammonia is liberated from ammonium sulfate. The liberated ammonia is trapped in boric acid, and that ammonium borate is back titrated with hydrochloric acid, which will give you the amount of ammonia liberated, and by using a conversion factor you can convert it into protein. So the standard conversion factors that we use for most of the proteins, we use 6.25. And if you look at the FSSAI quality standard, whenever a protein value is given, it will give you the conversion factor, that is multiplied by 6.25 or multiply by a different value. For milk, it is very high, and we generally have to multiply this by 6.38. So look into the conversion factor, look at the standard and what is defined in the standard. The other method of doing nitrogen protein analysis is to estimate the nitrogen by the original method, which is called as the Dumas method. The Dumas method is an instrumental method where the food is converted basically into carbon, oxygen, and nitrogen. So the first step in the Dumas method is to incinerate or break down the sample completely into its elemental runs. So here you have a furnace. So the steps involved is, the first step is combustion. So the furnace goes up to a temperature of about 950 degrees centigrade. The sample is completely broken down into its elements, and then the nitrogen that is released is passed into a detector, and the detector tells you the amount of nitrogen, and the nitrogen can be multiplied by the conversion factor. But not every laboratory can afford a Dumas instrument, but the Dumas method is becoming very popular. And then Coming to the last slide of it. So what happens if you do not take enough protein? So what is the result of it? Two major deficiencies of protein are noted, especially among the infants and children, and one is kwashiorkor. So this is a disease afflicting malnourished children. They have got swollen feet, enlarged abdomen, thinning of the hair, and dermatitis. These individuals cannot produce antibodies and can have lasting impacts of physical and mental growth. The other protein deficiency is that of marasmus, which is found in infants when weaning or when a mother's milk is reduced, and the symptoms are failure to gain weight followed by weight loss and emaciation. And these two figures clearly show the symptoms of thing. In individuals, protein deficiency is observed by what is called as muscle wasting. So you will find that muscle wasting occurs if you do not take enough of protein. If you take too much of protein also, you have you have to face the consequences, because the kidney can take only a certain load to remove the nitrogen from the body, and so you should not overload the kidney for clearance of the ammonia and the urea, which are the end products of the nitrogen metabolism or protein metabolism. So if there are questions, we will look. So the webinar is complete.